Hello, everyone. This is the online lecture to accompany Chapter 12, so I hope by the time you're listening to this that you have read the Chapter 12 book, taken your Chapter 12 quiz, and now you're ready to sort of go through it. If you look on the knowledge page for the course, there is also the corresponding slides if you want to follow along and take notes. I have the ability to draw and take notes as we go, so hopefully, see if that's working. Yep, that's going just fine. So hopefully I'll be able to do that as we go along here. So here we go, and welcome to Chapter 12 on translation. So as we have transitioned into molecular biology, I think one of the biggest challenges that arises is trying to understand how a molecular change in DNA actually leads to a change in phenotype. And we're going to focus on some of the examples that are pretty well known. And in fact, one of the first examples we knew of the actual molecular nature of the genetic disorder was sickle cell anemia. And so this is over 100 years ago. And here we had a physician who had a patient that suffered from fatigue and an enlarged heart. And when the physician took a blood sample of the patient, he noted that the red blood cells of the patient were sickled shape, hence the name sickle cell anemia. Biochemical studies identified that the hemoglobin levels in the blood were half of what we would consider a normal level. And so sickle cell anemia was really the first genetic disease to be understood on a molecular level. And today, given another 100 years of research, not only do we understand the inheritance of sickle cell and the phenotype of sickle cell, but we know the exact DNA base pair change that leads to the mutation. That DNA base pair change causes a change in a single codon. That change in a codon causes a change in a single amino acid. So it's a very linear type of genetic disorder when we talk about sickle cell, and we use it as an example. But just keep in the back of your mind that often the molecular changes we see that are going to disrupt or change protein structure and codons and amino acids are not always as clear exactly what type of change or how they might affect the way that the protein acts. So the other thing we have to get into today and think about is when we look at protein structure, we are now thinking about things in 3 dimensions. This will be one of the few slides I show this here for this particular chapter, but just the idea that we're going to start with the creation of a linear protein structure today, and that is the process of translation. That's adding amino acids from the N to the C terminal or the amino to the carboxyl terminal based on the codons on the mRNA. But just keep in mind that once these amino acids are added in a linear structure, they begin to fold. And that folding is dictated by the types of amino acids and the classes. We'll get onto that in just a couple slides here. You'll see it. But that folding, both intrastructure and interstructure folding as we get into quantity structure, is going to ultimately dictate function. So I have small children, as but no doubt by this point in the semester you're well aware of. So I often like to think of amino acids as Legos. So we have 20 different Lego types or 20 different building blocks, and what you can sort of think of is the number of types of things you could build if you had those different types of 20 building blocks. And when you start to think of all the different things you could build with Legos, you start to get an appreciation for all the different types of proteins that a cell can create and all the different types of function that those proteins can have. And we have some proteins that are very enzymatic in nature and help facilitate a particular biochemical reaction. We have other proteins that are very structural in nature, and sort of everything in between. But in general, each one of our 20 amino acids has common aspects to it. And so we have the amino group here on the left, which I'm trying to circle, and we have the carboxyl group here on the right. And you'll notice that when I'm writing out protein sequences and when I'm writing out amino acid sequences, that there's a polarity to it. Just like the 5' and 3' polarity we talked about in DNA and RNA, there's a polarity to amino acids. And so we go from the amino to the carboxyl terminal. Over here on the bottom where you see we have the R group, this is what varies between the 20 amino acids. And it's that R group that gives the amino acid its sort of chemical composition and ultimately predicts how that amino acid will interact with other amino acids, both within that same strand and within other proteins. All right, so each amino acid is specified by a codon, so it's 3 letters of mRNA that correspond to a single amino acid. And so we're going to talk about a lot of things in 3s today. Translation, for the purpose of this class anyway, always starts with AUG. For those of you that may have taken the C-phage course or other courses, we know that always is a dangerous word in biology. There are certainly other examples of start codons that we see throughout nature, but methionine and AUG is by far the most common start codon. And for the purpose of this class, we'll start all of our proteins with AUG and methionine. Additionally, translation always stops at one of our 3 stop codons. And so as we talk about the translation process today, the ribosome is going to be going 3 codons at a time, adding different amino acids until it gets to one of these 3 stop codons. And when it hits that one of those 3 stop codons, that's when we know we have hit termination and the end of translation will occur. All right, so a similar cartoon to what we just saw before. There are 20 different kinds or varieties of amino acids. They all have a side group here, and here they are sort of all on one single slide. Now, you can see the R groups are all down here. Oops. The R groups are all down here on the different parts of the amino acids. It is not my intention that you memorize or know all of the different chemical R groups for this particular class. It is my intention, however, that you know that there are 4 different classes of amino acids, and that type of class of amino acid is going to dictate ultimately how that amino acid will fold, interact, and eventually structure and function. So we have our hydrophobic amino acids here for number 1, our hydrophilic amino acids here for number 2, acidic, and basic. So oftentimes, and I think this is a good point to have this conversation, if you have a change in an amino acid in a particular protein sequence, predicting what that change will do, it's often helpful to start with a slide like this, right? If we change one hydrophobic amino acid for another hydrophobic amino acid, it's possible that that would have little to no effect on the function of a protein. However, if we would change a hydrophobic amino acid for a hydrophilic amino acid, now we're in an area where I would hypothesize that we might have some drastic changes, and we may have actually destroyed or sort of inhibited a lot of, if not all of the function of that particular protein. All right, so as our amino acids are coming together, this is almost sort of looking at our translation problem from the end result here. What you see is we have 2 different amino acids coming together. We have here the N terminus of the first amino acid. Here's the carboxyl terminus of the second amino acid. And so you get an interaction between the carboxyl terminus here and the N terminus here. And when they react together, what you get is a peptide bond. So just as we were talking about phosphodiester bonds in DNA and RNA, holding these strands together, in protein and translation, we have a peptide bond holding together the linear strand of the amino acids. Now, this structure we have here, as you see amino acids going one after another in a line, so this would be, amino acid 1 followed by 2, and this continuous type of thing could keep going on, so on and so forth. That structure where you're looking at all the amino acids in a line is known as the primary structure. So if you go back a couple slides, you can imagine what that looked like. All right, so number 1 here is usually the easiest and most obvious one for people to think about, but one thing I did want to highlight is it takes 3 different types of RNA molecules are involved with translation. So there are 3 different RNA molecules that are necessary in order for translation to occur. The first one, mRNA, is what we just finished on in chapter 11 when we talked about transcription, and this is the mRNA. So here we know it's eukaryotic because I have some introns that are being spliced out, the exons are being spliced together. And going from DNA to RNA was relatively straightforward because we were creating nucleotides from nucleotides. But now we're in a situation where we have at the bottom here our mature mRNA message. It has all of our codons in it, but we have to switch from being a nucleic acid to an amino acid. So really the molecule that's needed here is a translator, something that speaks sort of both languages of the 2 molecules. And that's where tRNA comes in. tRNA is transcribed just like messenger RNA, right? There is a promoter, there's an RNA polymerase that comes in and makes a single-stranded tRNA molecule. A couple of differences with tRNA is one, tRNA is not translated into anything. It is a non-coding RNA. It is functional by itself. tRNA also folds into a particular shape based on the complementary base pairing of that tRNA, and so it looks a bit like a cloverleaf here. And so at one end of the tRNA, down here at the bottom, you can see we have the anticodon. Those are the 3 bases that are going to be complementary of the codon of the mRNA. And then at the 3' end, where we have our 3' hydroxyl up here, this is where the amino acid is going to be added. And so when our tRNA gets charged, which we'll talk about in the next slide, then you have a molecule that has both the anticodon to recognize the codon of the mRNA and is carrying the appropriate amino acid to include into the protein molecule. And if you look at sort of at the top right here, you can see we have a picture of Francis Crick. You probably remember him from chapter 9 when we talked about the discovery of the double-stranded structure of DNA. Something I just wanted to bring up that he was also influential and sort of pertinently involved in the discovery and sort of the understanding that tRNA molecules must exist. All right, so just as we mentioned in the previous slide, these tRNA molecules, they are transcribed just like all other RNA molecules. They end up folding into a three-dimensional structure, and then they undergo a process known as charging. And charging is where we utilize some energy through ATP to add the appropriate amino acid onto the appropriate tRNA. And so once that charged tRNA is ready, now it can go and interact with the ribosome and the mRNA and be included into a primary structure of a protein molecule or a growing protein molecule. And lastly, what I want to mention for another type of RNA we have is ribosomal RNA. So ribosomes, these are the enzymatic workhouses of translation. We're going to talk about them in detail in a little bit, but they are made up of a number of different smaller molecules that make up 2 subunits. And approximately, if you sort of go by mass, half of the RNA is protein and half of the half of the ribosome is protein and half of the ribosome is ribosomal RNA. So you can see here we have the evolutionary sort of conservation of ribosomes. On the left we have prokaryotic ribosomes, and on the right we have eukaryotic ribosomes. So we have billions of years of sort of evolution between these two, and you can see how amazingly similar they are. I think this gives sort of some evidence and things you probably thought of already, but how important and how absolutely essential the ribosomes are. So any changes that have occurred in the last several billion years have often not been compatible with life. Now, one thing you can see is that both in prokaryotes and eukaryotes, there is a large subunit of the ribosome. So in prokaryotes, it's the 50S, in eukaryotes, it's the 60S, and a small subunit, so again, the 30S and the 40S. And those 2 subunits are going to come together to make the full ribosome. And unfortunately, for math purposes, when we have a 50S subunit and a 30S subunit, when it comes together, it makes a 70S ribosome. The reason that is it's not a measurement of mass, but a measurement of density. And so coming together is not quite a linear addition. Go from here. And then the last thing I sort of mentioned, in your human genome, you have hundreds of copies of the ribosomal RNA genes. So one thing I want to ask, and if we were in an in-person lecture day, I would sort of take a stop for it. So it might be a good point for you to pause and just sort of think about, why do we have hundreds of copies of this one particular gene? And so some of the things you may have thought of is, one, it's a very common gene. It's going to be needed by all cells. It's going to be needed in high abundance, so we have to make lots of ribosomal RNA in all of our cells. And another aspect of it is we can't live without it. And so having some redundancy built in and having some backup copies are probably something that's very favorable evolutionarily and to help our long-term survival. All right, so this is, I think, one of the most sort of comprehensive and beautiful slides when you think about evolution. And so what we think about here is the genetic code is conserved in basically all organisms that we know of, including some things that are not living. So viruses use the same genetic code as well. So the genetic code is essentially universal, is used by all organisms to translate messenger RNA into a polypeptide. There are 7 important features, and we'll sort of talk about these as we go through, but just sort of to put them all in one place. It is composed of triplet codons. It is non-overlapping and punctuation-free. We'll talk about what that means momentarily, but just imagine you're reading a paragraph with no punctuation and no spaces. It's degenerate because we have 64 different codons. We have only 20 amino acids, so more than one codon can code for the same amino acid. It is highly ordered, and that means that amino acids with similar chemical properties, so amino acids that are all hydrophilic or all hydrophobic, have similarly related codons. And so you could imagine from an evolutionary standpoint, this allows for a little bit more evolutionary wiggle room. And so mutations are likely, or more often than not, going to cause changes from a like to a like amino acid and not from like an acidic to a basic. And then importantly, we have to start and stop this thing, so it has start codons and stop codons as well. All right, so we have 20 amino acids. Why is it necessary that we code for each amino acid with 3 nucleotides? And again, I think this is a good point to pause and sort of think through the exercise, but why is it that codons have 3 nucleotides for our 20 different amino acids? All right, so I imagine you thought of something hopefully similar to this, but basically the reason is Goldilocks and the 3 bears, right? Goldilocks goes into the 3 bears' house and she eats the porridge, and the first one's too hot, and the second one's too cold, and the third one's just right. If we think about codons, it's a very similar idea, right? If we just had 2 letters of a codon, so you can see here, we only have 16 different combinations. That's not enough to accommodate all 20 amino acids. If we go to 3 base pairs codons, now we have 64 combinations. That's certainly more than enough to accommodate all of our amino acids, so there's no reason to go any larger than that. So it makes a lot of sense going that way. If experimentally you weren't sure of this, or this is your hypothesis, how is this something we could test? So let's take a second, think about that, and on the next slide I'm going to sort of talk you through a way you could test this particular hypothesis. All right, so what we're going to do is we're going to draw out this test, this hypothesis here, and talk about what they did. And so the way this hypothesis was tested, it was tested in vitro, which just means in a test tube. And so what they did was they used a repeat sequence over and over again in a test tube. And so for these purposes, I'm just going to write AUG over and over again. Okay. And so you can imagine if you put that into a test tube, and you put in our little mRNA here, and you put that into the test tube, having a little trouble with my pen here, then what's going to come out is a piece of protein or a polypeptide that's just a repeat of methionine over and over and over again. And what we're going to talk about here is something that's going to lead into sort of our next discussion in chapter 13. So let's say you took that same sequence, but you added one base. So before that first AUG, you wrote a C. And so, goodness. And so now we have that same sequence here. And so AUG, AUG. But now what we have is CAU, GAU, GAU, GAU. And so what we're going to get is a different peptide by adding one base. And now if we just add 2 bases, so let's add a U here and a C, now we're going to get UCA, UGA, UGA, UGA. So this is our +1, and here's our, all right, pen, +2. You get different peptides for +1 and +2. Now if we come down here and we add 3, and so let's just say we add AUG before it's added before, and we have AUG, 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 you're going to end up with the same peptide we had here. And so the idea is in a test tube scenario, when you add multiples of 1 or 2, you're going to completely disrupt what we call the reading frame, or the order of amino acids and how they're created in translation. However, if you add anything in multiples of 3, what you're going to do is you're going to keep the same reading frame so your peptide is the same. And experimentally, this is how they demonstrated that it was actually 3 base pairs in a single codon that caused an amino acid. All right, so as we've talked about before, when we've talked about replication and we've talked about transcription, is there's 3 key steps. You have initiation, elongation, and termination. One thing I did want to mention is we're going to go through translation today as it would happen in a prokaryote. And the only big difference I'm going to mention, other than the size of the ribosome, whether it's the 70S or 80S ribosome, is in initiation. There's a difference in prokaryotes and eukaryotes and how initiation occurs. Other than that, elongation and termination are both very, very similar. And then the other thing I want you to sort of be aware of is the enzymatic activity we have is peptidyl-transferase. And this is the enzymatic activity of the large ribosomal subunit. So both the rRNA and protein molecules in there are able to catalyze the formation of peptide bonds, the polypeptide that's being translated. All right, so big overview, and then we'll get into the different stages. So on the left is a typewriter. Typically, if you guys were in class, I would ask how many people have ever seen this or used this. It's becoming fewer and fewer every year. But if you can picture a typewriter, and not like the electronic kind, but the old school kind with the arms that came up and sort of hit the different letters on the paper and moved down the paper as you went, I think you have a really good image of what's going on during translation. And very similarly, what you can imagine is that the typewriter is now going to be our ribosome. And the mRNA molecule that we're going to have there is going to be our paper that we're reading from, and then we're adding in our protein based on what we're reading onto that mRNA molecule. All right, so let's start with initiation. Here you can see is our mRNA molecule. Here would be the 5' end right here. And what we have before we get to the AUG, so there's our start codon. You can see it right there. So before the start codon on the mRNA in what we call the 5' untranslated region, there's a sequence named the Shine-Delgarno sequence, named after the two scientists that discover it. And what it is that sequence here is complementary to a sequence on the ribosomal subunit. And so here we have the small subunit of the ribosome. The Shine-Delgarno sequence is complementary to there, and so those two sequences of RNA find each other. They create hydrogen bonds, and what that does is it sets up the AUG, or our start codon, in the right location on our ribosome. And so once our mRNA is bound to our small subunit of the ribosome, the appropriate tRNA, in this case it is methionine because we're starting at AUG. And in prokaryotes, we start with a modified version of methionine, and it's formulated. So you can see that we have the little F here for formal methionine. Is brought in by initiation factor 2, and then the large subunit of the ribosome comes in and sits on top of it. I sort of like think of initiation as building a hamburger, right? You have your small bun here on the bottom. The burger part of it can be your messenger RNA. Your toppings, your fixings are the tRNA with the methionine, and then the top part of the bun here is your large subunit of the ribosome. Now, before we go on and talk about how elongation occurs, I think it's important to notice when we're in the ribosome and both ribosomal subunits are together, there are 3 individual pockets or regions inside this ribosome that can accommodate tRNAs, and they're known as the E, P, and A site. And so, in general, A is for arrival. The only amino acid that doesn't arrive or start in the A site is the very, very first one. So this methionine here actually starts in the P site. But for every other amino acid we have in this peptide, it's going to start at the A site. The P site is where we catalyze the peptide bond, and the E site is ultimately where the amino acid, or the tRNA will exit after the amino acid has been attached. All right, so in eukaryotes, and again, this is going to be one of our places that we differ. The process is very similar, right? We start with a small subunit of the ribosome. Here we have the 40S versus the 30S, but it's the same idea. But in eukaryotes, there's no Shine-Delgarno sequence. What there is there's a 5' methylguanosine cap. And if you remember from our discussion in chapter 11 in transcription, that this cap gets added before the messenger RNA gets exported out of the nucleus into the cytoplasm. And so what happens is the cap binds onto the small subunit of the ribosome, and that allows the ribosome to begin scanning the messenger RNA. And again, we're going from a 5' to a 3' direction, but the ribosome, or the small subunit of the ribosome, is now scanning and looking for that first start codon. And so you can see it keeps scanning and going down the messenger RNA until that first AUG or methionine is lined up. Again, it binds the first tRNA we have here. The large subunit of the ribosome comes, and in both cases, you can see at the end of initiation, things look almost identical, right? We have an open A site in both the prokaryotic and eukaryotic ribosomes. There is a methionine sitting in the P site of our ribosomes, and our E site is currently sitting empty. The real big difference is how does the small subunit of the ribosome find that first start codon, and the difference is whether it's using the Shine-Delgarno sequence in prokaryotes or whether it's using the 5' methylguanosine cap in eukaryotes. All right, so as we mentioned before, we have these 3 different sites. Now ribosomes sitting upside down, but the same basic idea. We have our arrival, our peptide, and our exit site. I think they're easy to think about in sort of using this acronym here, or mnemonic devices, to try to think about not only what the 3 sites are inside the ribosome, but what's actually happening to the tRNA inside those sites. All right, so we transition now from initiation to elongation. So if you think here, our picture in the top left right here, this is pretty much where we ended at the end of initiation. And so what we have is an opening here at the A site. And what you can't see on the mRNA, but they've drawn in down here, is that there is a 3 base pair codon that's sitting on the A site right there. And so the appropriate tRNA, in this case it's alanine, comes in, the anticodon binds to the codon in the A site, and now what you have is the juxtaposition or close proximity of both the methionine that was brought into the P site and the alanine which was then brought into the A site. Once these two amino acids are in close proximity, we see the peptidyl transferase activity of the large subunit of the ribosome catalyzes that peptide bond. And now, if you remember back from slides here, we were going from an amino to a carboxyl orientation. And once that peptide is created, you can see we have that peptide bond there between the methionine and the alanine. The methionine is then released from its tRNA. All right, now what happens is you see a big shift. So I think this is a really nice way to think about the typewriter analogy. So we've created our peptide bond. We're ready to move on to the next codon. And so what happens is the entire complex shifts 3 bases. And so now you can see what was our tRNA in the P site is now in our E site. And you can see there's our empty tRNA from our methionine. The alanine tRNA, which had arrived in the A site, has now been shifted over to the P site. And we have a tethered growing polypeptide chain. So all these things are happening here. Importantly, what that's done though is opened up our A site. So again, we've moved down 3 bases on our mRNA, and now our next codon is ready for the next tRNA to come in and bind to it. And so that process repeats itself over and over again. And so what you're having is a repeating of the same process where the appropriate charged tRNA binds at the A site, the close proximity of the amino acid brought in on the A site with the growing peptide chain that's tethered on the P site, allow the large subunit of the ribosome to catalyze a peptide bond. And now you have a growing peptide chain. And once again, the entire thing has to shift. You'll notice one thing we didn't spend too much time talking about, but our methionine tRNA, once it's in the E site, it is able to dissociate and just leave the ribosome. So again, this is going to open up the E site for when we shift again on the next one. But this tRNA, what it's done is it's brought its amino acid, it's fulfilled its job, it can now be released in the cytoplasm. Truthfully, it will likely be recycled. It will be charged again and used to make another methionine amino acid in a peptide chain. And so here you can see the same process of the translocation. We've moved the entire ribosome 3 bases, again opening up the A site. And now what's going to happen is these steps are going to be repeated over and over again. So peptides or polypeptides in proteins can be quite a varying length, right? There are signaling peptides and certain hormones that are relatively short. There are other proteins that are thousands and thousands of amino acids in length. So the length or the time of this process is really going to depend on what type of protein is being translated. But these same steps of tRNA binding the A site, facilitating the peptide bond between the P site chain and the new amino acid in the A site, translocator shifting over 3 bases, is going to repeat over and over again until we eventually come upon a stop codon. So before we get over to termination, the stop codon, I think one way to think about it is just the 3 steps that happen during elongation. We showed that in picture form in the previous slide. Some people are more like words and helps them sort of understand things. So just as we highlight those, you get the binding of the appropriate charged tRNA to the A site. This facilitates the peptide bond between the amino acid chain that's on the P site and the new amino acid on the A site. And then that translocation is the movement of 3 bases of the ribosome, opening up a new A site for a new tRNA and amino acid and allowing the uncharged tRNA that has been released to exit through the E site. Now, on a molecular or biochemical level, this is what's happening, is all these amino acids are being connected in our primary structure as they get added over and over again. And again, I think one thing that's important is our very first amino acids over here, and to the left of it, at least the way we've drawn it, you can see our free amino terminal of that particular amino acid. And then our last amino acid has a free carboxyl terminal on the right side. So we still have some polarity and some orientation to our peptide structure here. All right, so termination is relatively straightforward. The process of elongation is going to occur over and over again. We've sort of highlighted a relatively long peptide chain here by the mark here. You don't know how many amino acids are in between this, but eventually, as we keep going on, we're eventually run into a stop codon. Now, there are no tRNAs and amino acids that are complementary to a stop codon, but what there is a release factor. And so the release factor comes in, it binds to the stop codon at the A site, and now what happens is there's no amino acid here to connect the chain to. right? And so we're going through sort of the same process, but there's no amino acid for the peptidyl transferase to connect the chain to and keep it tethered to the ribosome. So what happens is when the translocation occurs, the protein's released. And so as we have translocation in the past, it was still tethered ribosome, but when we have our release factor there and when we're on our stop codon, the protein's released into the cell. Oftentimes it'll go through more folding and post-translational modifications. And what we have then is the dissociation of the ribosome. So the two subunits of the ribosome come apart, the last tRNA is released, the mRNA comes apart as well. Now, all of these will be reused again, right? A single mRNA molecule is transcribed, and it can lead to the translation of many versions or many copies of the same protein, depending on the needs of a cell at a particular time. But this is the end of the translation of that one particular polypeptide. All right, so again, translation is pretty straightforward. We have the binding of the release factor of the codon, the release of the peptide during translocation, and then eventually the 7DS ribosome is dissociated and recycled inside the cell. We've used, again, prokaryotes as an example here. This process would be very, very similar in eukaryotes, other than the size of the ribosome and some of the name of the proteins helping facilitate it. But for the purposes of this class, we're going to focus on the prokaryotic translation. All right, so I think this is a nice image to look at here. As you're studying, as you're working on this in groups and with other people, I think if you can sort of talk through those 3 steps, draw your ribosomes, draw the peptide chain extending, you'll be in very good shape, and something sort of a nice drawing to try to get to there. All right, so ultimately what we want to be able to get to here as well is try to talk about how we use the code and how it's determined. They did a lot of in vitro experiments, just like we talked about before, where they were putting artificial mRNAs inside a test tube and putting in ribosomes and all the amino acids and all the tRNAs and energy, and basically asking what type of amino acid was created. And so what we have is ultimately a genetic code that looks something like this. Now, the way you use this is the first letter of your codon. This is the second letter up here. And then the third letter is on this axis over here on the right. So if you look in the middle here, what we have are 64 different combinations of 3 letters of RNA. So let's start with AUG, as that's something we're very familiar with. That's going to be our start codon. And so our first letter would be here, A. Our second letter would be up top for U, so we find that current box. And then G is our last one. As you can see, as we look right here, there is our AUG. That is our initiator molecule right there. Now, I will say for this particular one, and this is the image I believe you have in your textbook as well, the stop codons, they all have different names for ochre, amber, and opal. We're not going to use those specific names, but you can see they're named here as terminators. So that's the same thing as a stop codon. So if you were translating a particular mRNA message and you came across UAA, UAG, or UGA, you'd know that that would be the termination or the end of that particular peptide. So I think it's good to practice with this type of molecule here. I have a couple slides in the next to practice with. I'm going to go through and try to draw in here as best I can what those are. If it doesn't work, I may have to leave it as a separate document online. But what I would encourage you to do as we get to these different examples, and here you can see we have 3 different examples of mRNA molecules I'm going to ask you to translate, is pause the lecture. Right? Try to go through it yourself first, see if you get stuck anywhere, and then go back and watch how it's done. I think it'd be much more effective that way. So if we just start here with our first one, and so you can see here is our first mRNA molecule, and so I'm going to start by creating the peptide that would be associated with that. And so again, I'm going to make my N terminus first because I want to go there. My very first codon is AUG. So again, I use my chart. I go to my A, my U, and my G, and I know that's methionine, so I'm just going to write MET for methionine. Now, there's no punctuation, there's no skipping codon, so my next codon would actually be the next 3 letters here. So in this case, it's AAA. So I come back down here. Here's my first letter A, second letter A, and third letter A. And so you can see what here it is lysine. And so I'm just going to draw this here as I go like this for lysine. My next one is going to be CCC. So I come over here on the chart again, and it looks like it's proline. GGG is next, and you can see I'm sort of underlining them as I go, so I make sure I don't do it, one of them twice. And if I come all the way over here, this looks like it's going to be glycine. I come back to AAA, And so I know that's lysine, so I'm putting another lysine in here. And I think this is a good sort of discussion point. If I look at my next amino acid, or sorry, my next codon, we go back to AUG. So I know from the beginning that that's methionine. Is it possible to have a methionine in the middle of your protein? And the answer is yes, right? Methionine starts our proteins, but it's also one of our 20 amino acids. So methionine can be right in the middle of our protein there, and we just add it right in. Our next one is UUU, which is phenylalanine, which I'm sort of drawing all over right now, but hopefully you can still see it. And then lastly, the last one we see here is UGA. And now if I come over here, that's my terminator codon. So I think one of the things that's easy to forget is people will often write stop in here or terminator. It's important to remember that a stop codon exists in RNA. A stop codon does not exist in protein. There's no such thing. And so what that would mean is if I ran into my stop codon here after the UUU, that this phenylalanine would actually be the end of my protein. So what I've done there is I've added a C just to indicate the carboxyl terminus or polarity of this particular protein, but that's what it looks like. And so you can see my handwriting is absolutely horrific, but that's the kind of idea and the kind of way you want to go through it. And so if you keep this here or keep that one going, the next thing you can see is we have different mutations. So what I've done is I've added a frame shift. So I've highlighted it in red here. You can see there's an A that's been added to this particular protein. So if we go through this one just to translate it, what you can see is, again, I'm going to start with my N terminus. My AUG, that's methionine. My next amino acid is AAA, which is, oops, there we go, lysine. And now I run into trouble because previously it was CCC, but an A has been added. So now my codon is actually CAC. So I have to look up a completely different codon right here. And so if I look over here, that's actually histidine. And then because the code doesn't skip any base and it doesn't overlap, my next codon is actually CGG. And so you can see that I've disrupted this entire sequence here. And if I look over here for CGG, I believe I'm looking at arginine. And then I have GAA is next. And so we're going through this, and you can see what I've done now is I've completely disrupted not only the codon in which I added a base, but I've disrupted almost the entire protein sequence. So now I'm on AAU. I'm trying to go above and below so you can sort of see it as I go. So for AAU is here. And then GUU is next. And so if I come down here to GUU is valine. And then I get UUG, which is here. And then often I get this question of, what do I do with this extra A? So we didn't get to an actual stop codon here, right? We ran out of mRNA before we found a stop codon. If this were to happen in an actual cell or in a test tube, eventually the ribosome would just fall apart, right? There's nothing left for it to do. So what we do in this case is we have no way to translate this A. It's not a 3-letter codon. It's a single base pair one. So this becomes the end result here of our particular amino acid. Now, if you look at that amino acid sequence of methionine, lysine, histidine, arginine, and so on, and we go back a slide and you compare it, you can see not only did I disrupt that one particular amino acid and where the mutation was, I've now actually disrupted every single amino acid afterwards. And this is sort of leading into a nice discussion of our next chapter, which is chapter 13 on mutations. But this is a frame shift mutation, so it's shifted the entire reading frame of the protein. Frame shift mutations often have incredibly large-scale phenotypes, right? There's a very little chance this protein would work in the same way that it worked before, if it even works at all. It may just be a non-functional mess. All right, let's do one more. And here I haven't had a frame shift, but what I have is a point mutation. And so you can see here where there was an A, there is now a U. And so if I go back and I'm going to translate my nonsense mutation now, so again, I'm doing this one down here as a comparison. Again, I start with methionine. My next amino acid lining up here is going to be lysine. All C's is proline. All G's is glycine. And then you can see my next one is UAA. And if I come over here, that's actually a stop codon. And so what I'm going to have here is that's the terminus, or that's the end of my particular protein sequence. Because there was a stop codon there, the ribosome would fall apart. Now, a couple of things to think about. One is, it just turns out that for this example, the next amino acid, or the next codon coding for an amino acid is another start codon. Can I start again? And the answer is no. Because if you go back and you think about the initiation of all this, we had to have, if it were in prokaryotes, a Shine-Dalgarno sequence. And so there's no Shine-Dalgarno sequence here to set up the ribosome properly. or for new carrots, we had to have a 5' methylguanosine cap. And so we don't have any way to start it here. And so what you see here in a nonsense mutation, and this is the type of point mutation that changes a codon for an amino acid to a codon for a stop codon, what you can see in this particular example is called a nonsense or sometimes a truncation mutation. We haven't changed any of the amino acids in the protein, but we've made it a lot shorter, and we didn't exactly code the entire thing. Now, what a truncation or a nonsense mutation is going to do in an organism really depends on which protein it is and where the truncation happens. If it happens very early in the protein, it probably is not going to be functional at all. If you have a protein that's thousands of amino acids and it happens all the way towards the end, maybe it retains some of the function. It's very, very much context dependent. All right, so one more topic I want to touch on here, and as we go forward, and it's a really, I think, big picture question. What is a gene, right? Gregor Mendel, and sort of how we started this semester, Gregor Mendel would have called a gene one unit of inheritance, right? We now have a little bit more definition as we went through transcription, right? A gene has a promoter. It's some sort of region of the DNA that codes for a particular mRNA. But around the time that they were discovering and studying translation and looking at the different codons that we're making, there's a lot of discussion about what is a gene and how can you define a particular gene. And so there's an absolutely seminal experiment here from Beadle and Tatum in which they concluded, and so we'll sort of get to the conclusion first and then talk through how they did that particular experiment. One gene coded for one enzyme, and we'll update that in a few slides, but this was sort of an absolutely brilliant and seminal experiment. And so what they were studying was a model system known as conidia. And so like all good geneticists, they mutated conidia to study a particular phenotype. And so in this case, they're creating their mutations with X-rays, right? There's a lot of different things they could have done. And what they did was they took each one of their mutants and mated it in complete media. Or sorry, grew it in complete media. So each one of these tubes represents a different mutant. And complete media is basically, as we talked about in chapter 8 when we were talking about prokaryotic genetics, it's not only a sugar source, but it's all of the nucleic acids, all of the amino acids, anything that normally a conidia would be asked to make on its own, all of that's provided for it. So what they were looking for is mutants that could survive on complete media. So they needed mutants that could do two things. One, survive on complete media, and then two, not survive on minimal media. So minimal media, much like we talked about with prokaryotes, is just the minimal requirements for a wild type organism to survive. But what's going to have to happen is that organism has to create or synthesize all the amino acids and nucleic acids it needs for survival. So Beadle and Tatum did this screen, and they were basically looking for mutants that did two things. They could survive on complete media, but not survive on minimal media. So if you're thinking about this in your head, really what they're asking for is they're looking for mutants that had a growth deficiency or a growth requirement. And then what they did was an absolutely brilliant set of experiments. There's nothing complicated about this, but the amount of work and labor it would take is kind of amazing. And so for each one of their mutants, what they did was they started growing it on media, but adding things back one at a time. So here you can see, if we go down here, they've taken one of our growth mutants, and we've grown it in minimal media, and it's controlled. You can see none of the conidia growing, so they're dead. We've done minimal media plus amino acids, and then they came back to life. And so what they concluded was that the growth requirement here was an amino acid, or at least one amino acid. Because, and the way they concluded that is when they gave this particular mutant amino acids, they were able to go back and survive. And then what they did was they ran 20 different vials on the next set of experiments. And so each one of these vials here down in E is minimal media with one amino acid brought back. And so for this particular example, what you can see is none of this particular mutant strain survives except when they give it arginine. And they ran this exact same type of analysis on hundreds and hundreds of different mutants. And what they found was something very consistent, was that each one of these mutants had a single growth deficiency. So it was deficient for either one amino acid or one nucleic acid. And their mutants had deficiencies for different things. So it wasn't like every mutant was deficient in arginine. Some were deficient in arginine, some lysine, some serine. but each one of those mutants was deficient for one molecule only. And so what Beadle and Tatum hypothesized, and correctly so, is that a lot of these amino acids and nucleic acids are created through the synthesis of molecular or biochemical pathways, and that each one of these pathways is required or necessary on a bunch of enzymes to work. And so what they concluded was, since they're mutating genes and causing a mutant phenotype, and that mutant phenotype was a growth deficiency, that each gene must code for one enzyme. So that's where their mantra of one gene, one enzyme must be coded for. Now, we've updated that today for one gene, one polypeptide, but I still think it's a really good way to start thinking about the molecular nature of genes, right? And the reason we had to update it to one gene, one polypeptide is there are certainly examples of genes and there are certainly examples of proteins that aren't enzymatic, right? Histones is an example of one. That's a very structural protein. Still a gene, still makes a protein, but that was the idea, and that's how they went through everything. All right, so that was everything I wanted to say on Chapter 12. I hope that was something that was clear to you. Again, I encourage you to go back, read the book, come to office hours, ask questions, make sure you do the problem sets, and I appreciate you sort of sticking in with our online lecture today. All right, goodbye, everybody.